In these next sets of videos, we're going to do the reproductive system. And in this video, we're going to focus on male reproductive system, and the next one we'll do female reproductive system. So the overview of the reproductive system is that we have our primary organs, and they're going to produce these cells called gametes. Gametes are just half of our genetic information that when an egg and a sperm come together, they'll combine to be the full complement of genetic information. If we didn't split our genetic information in half, what would happen is as generations went on and on and on, there'd be so much genetic information that cells would have to get bigger, and there'd be just tons of redundant information. Plants sometimes do this, where they have tons of redundant information, and their chromosome numbers get bigger and bigger and bigger, and that leads to some weird problems. So with us, we basically take our normal complement of genetic information, 46, divide it in half, and then in each of us we produce one chromosome, which is going to lead to sex, which will lead to later an X and a Y. If you have two X's, you're going to be female, and if you have an X and a Y, you're going to be male. And depending on if you have the two X's, it's going to lead you to have female characteristics, which is going to be a uterine tube, a uterus, a vagina, and the ability to have a uterus for nourishment of the fetus. If you have an X and a Y, that's going to lead you to make ducts and glands and the penis for delivering sperm. With the male reproductive system, its function, of course, is to produce sperm, produce sex chromosomes. Those are going to be the X's and the Y's. The male is really the only one that dictates if the person is going to be male or female. The female only gives an X chromosome. And usually what happens is the Y chromosome is super small. It's kind of really small, and the X chromosome is much, much bigger. So when these guys divide into different sperm, what happens is the one with the Y chromosome, because it's so much lighter, because it's got such a smaller chromosome in it, will swim faster. And that's why we see an initial birth rate of a lot more males than females. I believe it's something like 52 to 54%. And then eventually what happens is, because males don't have the same immune system that females have in the beginning, females actually survive a lot longer to fertility. So we'll go ahead and see that the roles will almost reverse, where males will start to die off and there'll be a lot more females. The males have testes, and how we break the testes down is going to be that we start out with these small tubules that are going to start to break normal cells down and break their genetic complement down. So we have 46 chromosomes, and what's going to happen in these little areas are that we're going to start to break that material down to 23 chromosomes, and that's going to be the initial phase of making sperm. So what we're going to do, we're going to start out in these semi-fairness tubules, which are going to drain into the reticestals, which is here where you can see that they all sort of start to group up. And eventually what's going to happen is they're going to start to go, eventually, this is going to be the epididymis, where sperm will mature. What you're seeing is the kind of cells that would be here are called Sertoli cells, which are a nurse cell. And they're doing this thing called spermatogenesis, and they're basically taking 46 chromosomes and dividing them into 23, and eventually helping us make sperm. If we look at the testes itself, so here was the testes, here's that epididymis that we talked about, and eventually what's going to happen is we have that vast difference. This big tube is the spermatochord. The spermatochord is going to go up, and it's going to sort of tuck back into this thing called the inguinal canal, and then turn around to get to the front of the penis, or the back of the penis, which will lead eventually to the front of the penis. And what happens is we have a few different things in the penis that helps us, or in the testes, that help with temperature of the testes. It turns out that we have this cremaster muscle, which is this kind of webbing that will go all around. You don't really see it. You see a little piece of it here, but it's kind of like this webbing that goes around the testes. And they're designed to pull the testes closer to the body when the body gets colder, or the testes get colder, and push the testes away when the body gets warmer. And it's really designed that sperm is designed perfectly at 35 degrees Celsius. And our internal temperature is 37 degrees. So it doesn't really make it for a good environment for sperm. So we have to have the testes outside the body so we can have it a little cooler. And as our body gets hotter, we're going to pull the testes close to it. As the body gets colder, the testes get colder, we're going to pull the testes closer to the body. And of course, when the testes get too hot, these cremaster muscles are going to loosen and try to get the testes away from the body. So that'll be done by the cremaster muscles. We also have these dartus muscles. They're smooth muscles and they reduce surface area. That's part of the wrinkling on testes that you see, and that's designed to get rid of heat. Because what's going to happen is all these arteries and veins surround the testes, and as the testes get hotter, they start to circulate blood faster around the area. And what they're designed to do is basically act like a radiator to get the heat away from the testes so we can continue to make sperm. If not, if we get too hot, we won't be able to make sperm. It's got to be at that 35 degrees temperature. So we're going to constantly be moving this around. And how we're going to get rid of that heat is going to be through the venous plexus. And that's just going to be, again, just a way to get rid of heat. So we're going to have more veins there, and veins are going to be able to move the heat away from the testes. And just here's a little more of the countercurrent exchange. As the testes get too cold, we're going to pull it up to the body. If the testes get too hot, we're going to just pull it away. And testes ideally want to be at 35 degrees. If we look at the production of sperm, you'll see that we have an efferent ducts, and these are basically where we're going to start to get all of that sperm to sort of, these are actually at this stage, they're just cells. And they're going to start to go down to the epididymis. We saw a little bit of the epididymis in the cadaver. And that's going to be the site of sperm maturity. At this stage, this is where it gets that tail, and it gets a couple of chemicals that gets added to it, so it makes it more viable sperm. It takes about 30 days, 25, 23 days to go ahead and make a cell go from 46 chromosome to eventually a viable sperm cell. The vas deferens is going to be the passageway away from the epididymis, and that's going to go to the ejaculatory duct. And the ejaculatory duct, if you see, here's the vas deferens, we're going to be up over here, and you're going to see that we're going to share the same area, the same tube as the ureter up here. And and here's the prostate, and you're going to be able to see that we'll be able to push that sperm through this elaborate couple things that has to go on. So the difference between the vas deferens and sort of a garden hose, little stupid joke, is basically that both of them are going to share the urethra. But to share the urethra, we're going to have to add a chemical, and that's going to be a pre-ejaculant, and that's designed to get rid of any urine that might be in the urethra. Urine has a lower pH, sperm has to have a really specific pH, so that pre-ejaculant is designed to clear out any urine, neutralize the pH, not just in the male, but also in the female. So that pre-ejaculant is kind of like a material that sperm can sort of then come and go ahead and have the right pH. The epididymis is going to collect that sperm and eventually propel it through the vas deferens. And what a is where they just cut that vas difference. They used to tie it, just kind of like what you're seeing here, but what would happen is sperm would still sort of come out and swim and find the other way. So now what they're doing is where they'll go ahead and just sort of cut the tubes, and they'll hit it with a little bit of heat to close both sides of the tubes, and it'll make it so it's a lot more effective. What they can do to reverse it is they'll take these two pieces and join them back up, cutting that little area that we've singed with heat, and now we can go ahead and even add a vein to the outside, so we can kind of repair that, or we can give it some time to sort of heal. So reverse vasectomies are a possibility now. If we keep looking at some of the secondary organs that we have, so here's the testes, here's the epididymis, here's the vas deferens, and the spermatocord is what the cord would be in. This would be the inguinal canal that you can't really see here, but you can see it with the cadaver. And then we could start to look at our first sort of organs that we need to see. We're going to go ahead and have a prostate, a bulbar urethra, and these glands are going to be helpful in helping us create the liquid that sperm has to travel in as it goes down the ureter into wherever, the female. So the secretions that have to be in sperm are a couple things. They are going to go ahead and have to have, sperm itself is going to have fructose in it, and the fructose is designed to be the sugar that sperm could bring into it to keep itself moving. Sperm has to travel the equivalent distance, you know, if you kind of made yourself bigger or made them smaller, from LA to New York. And that requires a lot of energy, and that's going to come in the form of fructose. I'll go through the rest of the material for this in our next video.